Thank you very much, Christian. So I'm going to present you the GEroNIMO project, which started 2 years ago. So we are moving on chicken and pigs. So bye-bye aqua fish. So. There are two parts in this presentation. One part on context and aims and work package organization of the project, and another part more focused on molecular aspect in relation to this session. So, as you know, we have to face big challenges as there are mice. Yeah. As human population grows, environmental degradation, food security, changes in social and cultural values. And in this context, the aim of GEroNIMO is to rethink animal breeding in pig and chicken to move toward a more sustainable model, ensuring first production, but also promoting efficient resource use, animal resilience and welfare, and preserving genetic diversity. So a lot of aims. The strategies that we chose to do that was to use current omic technologies associated with multiple phenotypes related to production in terms of quantity or quality of products, feed efficiency, productive longevity, in particular in laying hens, fertility, resilience, and welfare. And this to contribute to try to improve knowledge on genome to phenome relationship, accounting for both genetics and non-genetics mechanisms controlling traits. So the GEroNIMO consortium is involved 21 partners from 11 countries with 3 private companies in pig and chicken, 3 professional associations as well in pig and chicken, experts in innovation and project management, and a lot of academic experts in different scientific fields. The GEroNIMO is organized around 8 work packages with 5 scientific work packages, 3 in blue here, in which all the omics data are generated, and this data has the fuel to the 2 other scientific projects in green here. In the P4, the aim is to develop methods based on genetics and epigenetics component of traits for improving selection strategy, like a lot of other projects. And in the P5, the aim, there are different aims, and one of them is to analyze the societal, ethical, and legal issues related to genomics innovations by using surveys. And another aim is to analyze the feasibility, the risks and benefits of genome editing by performing simulation studies, and also of epigenetic editing by an experimental pilot study conducted by Guillaume. I don't know if he's in the room. So let's go to the 3 work packages in blue, in which all the omic data have been generated. So as you know, the phenotype variation is determined by the gene expression regulation, which is under the control of genetic variants for one part, and for another part to the epigenetic modification, which are the result of environment. So based on this idea, the aim of the P1 and P2 is to identify biological mechanisms affecting trait variation, considering genetics and epigenetics component. So in the P1, we have quite large animal design to perform quite efficient GWAS analysis related to different scientific questions, for example, about sperm quality and growth and feed efficiency for pigs, and about laying weight, egg quality and feed efficiency during long careers over 90 weeks for laying hens. And here, just a few results about this double P1. Double P1. You can see here a lot of QTL which have been located by different people for daily gain, back fat thickness in different breeds of pigs by using 20,000 genotyped pigs per breed, or about semen concentration by using 400 genotyped pigs, or in laying hens, for example, QTL for body fat reserve. And for all these QTL, which are plenty of good gene candidates. In the double P2, we have smaller animal design compared to the double P1. Yeah? And the aim is not to perform GWAS analysis, but just to compare epigenetics and genetics variant between different environments. And these environments are different transport and rearing conditions here in chicken only, between different diets or different ambient temperatures or seasons or different ages and generation for pig and chicken. So a lot of experiments in this work package led by Tatiana, which is here. Who is here? Sorry. In the double P3, one of the aim is to map genetic and epigenetic variation in different chicken and pig local breeds to propose strategies for optimizing the conservation of genetics and epigenetic diversities. So after 2 years, we have accumulated a large number of animals, of samples of omics data, more than 90,000 genotypes and phenotypes in pigs and chicken, around 2,000 transcriptomics data, mainly poly-RNA-seq, but also small RNA-seq and ribodepleted RNA-seq for some samples. And coming soon, around 10,000 epigenomic data, so it's a methylome data, with associated epigenotypes. And with this omics data, we perform genetics and epigenetics analysis like GWAS, allele-specific expression, EWAS, like in another European project, to try to decipher the part of genetics and epigenetics involved in the variation of phenotype. About the pipelines that we used in GEroNIMO, we use pipeline from NF-core, which ensure the reproducibility of the analysis when they are available, like NF-core RNA-seq. Otherwise, we use in-house pipelines combined with pipelines already approved by others, in particular other scientists in human. For example, TensorQTL from the Broad Institute for eQTL analysis, or FASER for allele-specific expression. About the genome assembly, we use the last version available on the website, Ensemble website, mentioned here for pig and chicken. And about the genome annotation, so the gene model in the genome, we use for pigs the version 1007 from Ensemble, and an improved version for chicken, combining gene models from different resources, including, of course, Ensemble, as shown just after. So I'm going to finish the presentation with some slides about this new improved version. So we successively gather 6 gene model resources I don't have time to give some details about the methods, but these 6 resources were the 2 reference genome annotation, NCBI, WebSeq from NCBI, and Ensemble from MBL EBI, including gene switch RNA-seq data. The 2 resources related to the pilot FAANG chicken multi-tissue RNA-seq data from FRGencode and the University of Davis, and 2 additional resources from INRAE and NonCode, which is dedicated to non-encoding RNA. And using this strategy, we increased a lot the number of protein-coding genes in blue here in NCBI or here in Ensemble to 24,000 in the new atlas, and the non-encoding RNA loci in red here to 44,000 loci. And we then tested the reliability of these gene models by quantifying their expression across 47 tissues. For that, we analyzed more than 1,000 RNA-seq samples from different data sets, well chosen to represent a good variety of physiological system in chickens. And we found more than as you can see here, more than 90% of protein-coded genes in blue here and 80% of non-encoding RNA, which were expressed in at least 1 tissue, showing that the models of this new atlas are real. And finally, in order to easily work on this gene-enriched atlas, we created GEGA, a user-friendly tool, which is accessible on this URL, which gathers functional and genomics information. And for example, if you are interested in a QTL region, like in GEroNIMO, you can define this region quite easily, like here for the body fat reserve QTL on chromosome 2, and display very quickly all the genes present in this region. So here, for example, there are 17, thank you, 17 protein-coding genes and a lot of non-encoding genes. And you can then get various genomics and functional information related to these genes. For example, you can get the functional annotation like GeoOntologies with search here function, and select the genes with ontologies that are consistent with a trait of interest. And for example, here there are 2 genes, these ones, which are related to fat or lipid terms related to the QTL of interest. And you can also get expression across the 47 tissues with the possibility to display graphical views, like you can see on the right, and for the 2 genes previously selected. And you can see here that it's possible, if you want, to prioritize, I don't like this verb, the genes expressed in a tissue that is consistent with a trait of interest. And in our example, CYP5A is highly expressed in the liver, which is a key organ of lipid synthesis, making these genes a good candidate for this QTL. And you can explore the region further by obtaining more information about, for example, genomic orientation between 2 neighboring genes, like protein-coding gene and a non-encoding gene, which can be a regulator of the protein-coding gene or orthologous gene between mouse, human, and chicken, or co-expression, etc. So I would like, in conclusion, we think that this tool should be useful for the GEroNIMO project to explore the multiple QTL regions that we have located in chicken. And I would like to thank people who contributed to this atlas and this tool, and in particular my PhD student, Fabien, who is here, and Philippe Bardou, a colleague from INRAE. And it's too late to see the poster because it's removed. So, and thanks a lot for your attention.